Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled Reconstruction of Disease Pathology for Drug Discovery, Focus on Respiratory Diseases, Asthma and COPD. The American Lung Association estimates that almost 26 million people in America suffered from asthma last year. The organization also calculates that 13.1 million Americans had COPD in 2009. And the Asthma Society of Canada estimates that more than 600 million people worldwide suffer from COPD. Both asthma and COPD create a huge and costly health problem for many countries. Better methods of treatment are sorely needed. Today, our webinar panelists are going to talk about some new ways of thinking about these diseases, and they will suggest some innovative approaches to studying these disorders and developing more effective therapies. Let's meet our panel. Dr. Ian Adcock is Professor of Respiratory Cell and Molecular Biology at the National Heart and Lung Institute, Imperial College London. Ian is going to talk about the key challenges in asthma and COPD research. Dr. Scott Tebbett serves as an Associate Professor at the James Hogg Research Center at the University of British Columbia in Canada. Scott will explore the use of blood as a resource for the molecular investigation of respiratory diseases. Dr. Ian Nikulski, Vice President of R&D at Thomson Reuters, will discuss MetaMiner, a novel pathway analysis platform focused on asthma. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So, if everyone's ready, let's get going. Dr. Ian Adcock will be our lead-off presenter. Ian?
Okay. Good afternoon, everyone. I'd like to start my presentation by showing a slide that's obviously slightly out of date from our earlier introduction. We think there are well over 500,000 people worldwide, and maybe even closer to 750 million people worldwide who suffer from asthma and COPD. In fact, COPD is one of the few chronic diseases that's increasing in incidence worldwide and is expected to be the third major cause of mortality within the next decade. Even before patients come into the clinic with asthma, there are acute changes occurring to the epithelial cells within the airways in response to allergen challenge. So we have epithelial denudation and subepithelial fibrosis occurring. Activation of the epithelium by the allergen will cause mast cell degranulation and recruitment of pro-inflammatory factors and also bronchoconstrictors. We also see activation of professional antigen-presenting cells such as macrophages and dendritic cells, which in turn can drive the classic Th2-mediated eosinophilia that we think are important for driving the asthmatic response. The release of eosinophil products causes changes in airway hyperresponsiveness and vasodilatation angiogenesis, so remodeling of the airways. So this is a classic paradigm of how asthma is driven. So why do we need new therapies? Most patients with asthma respond well to current combination therapies. In fact, even patients who have well-controlled asthma on existing therapies may also benefit from having better therapies because they become more compliant. Compliance can also be improved by developing maybe safer oral versions of the current treatments we have, or maybe new, more efficient treatments could become available. One of the major problems we have with current treatments is that they do not alter the course of disease or provide a cure. And this is one of the major aims for therapeutic development over the next 20 years or so. Numerous researchers, both in academia and in industry, have looked at a number of different approaches to try and target the classical Th2-driven eosinophilic process that drives asthma, and a list of potential targets in drug development is shown on this slide. However, many of these have not proved as effective as we would hope from the models we have both in vivo and in man and in animals. And this may be because it's now become very clear that asthma is not a single disease. Rather, it is a syndrome of many different subtypes of asthma, and these subtypes differ both in adults and in children. So if we try to pick one particular drug that could target many of these different phenotypes, without selecting patients who are likely to respond, it's not surprising that many of these new expensive drugs have failed in clinical trials where we have not pre-selected patients who are likely to respond. One way of trying to define these phenotypes of patients is to take clinical phenotypes such as lung function, age of onset of disease, and see whether or not we can use this data to cluster patients into separate groups. And this is data from Wendy Moore and the Severe Asthma Research Program, which is a group of researchers across the United States who are trying to examine severe asthma. And they were able to cluster asthma into subsets that were loosely defined upon the lung function, but had other qualities that went across the whole spectrum. So we see some severe asthmatics would fit into cluster associated with current GINA guidelines, and others would go right across the spectrum to even mild, moderate asthmatics would have some degree of steroid insensitivity, for example. So where we see clustering can start to subdivide these phenotypes of patients. In another approach, Ian Pavord and Chris Breitling in Leicester have looked at sputum eosinophils to try and separate out groups of patients who may have highly responsive disease or a disease that's less responsive to inhaled corticosteroids, for example. So most patients will have an atopic early onset disease which responds to conventional therapies, but there are other subsets of patients who may have discordant inflammation, so high levels of sputum eosinophils, but with very low symptom scores. Others will have high symptoms and low eosinophils. By trying to understand these subtypes of patients, it's been possible to take patients with high levels of sputum eosinophils and use this to subphenotype patients who are more likely to respond to Th2-driven eosinophilic responses. And such an example would be an anti-IL-5 drug. And here is some data from the Leicester group shows the effect of an anti-IL-5 mepolizumab on the number of exacerbations that occur over a year compared to patients on placebo. So these are patients with high levels of sputum eosinophils despite high-dose inhaled corticosteroids. And you can see a much greater improvement in exacerbation rate in the patients with anti-IL-5. This clearly demonstrates that picking a marker of disease can predict whose biomarker will be predictive of who's more likely to respond to a particular drug. This data has been followed up in a subsequent much larger study which has recently been published, where it suggests that maybe blood eosinophils may be an even better predictor of the response to anti-IL-5 therapy. In another approach, Prescott Woodruff and John Fahey in San Francisco have used a transcriptomic approach to look at the epithelial cells from patients with asthma and to try and delineate a fingerprint or a signature that would tell them who's more likely to respond to inhaled corticosteroids versus those who are unlikely to respond to corticosteroids. And they're able to pick out a signature that relates to Th2, the classic asthma phenotype, who are the steroid high responders. And they found that if they looked at lung function, it was possible to separate the patients who responded very nicely to inhaled corticosteroids. In this case, on the panel on your top right, the fluticasone response was much, much higher in the patients who had this conventional or this classic Th2 signature compared to the lack of response seen in the patients who had no Th2 signature. So again, we're using a biomarker to try and define patients who are likely to respond or not respond to particular drugs in asthmatic patients. So this led the same group of researchers to look at the ability of anti-IL-13, which drives the presence of periostin and other Th2 markers from epithelial cells, to define patients who are more likely to respond to therapy. So here they looked at patients who had high levels of periostin versus those who had low levels of periostin and could find a much greater improvement in lung function after anti-IL-13 therapy in the patients with high levels of blood periostin versus those who had no improvement in lung function if there was no periostin or low periostin levels in the blood. So again, suggesting that taking biomarkers of patients with a particular subphenotype is able to define the patients who are likely to respond to novel therapies. In Europe, there's a large consortia of over 30 academic groups and 10 major pharmaceutical companies who are taking a larger approach to try and subdivide asthmatic patients into subphenotypes of severe asthmatic patients, where they're trying to combine not just single omics data such as transcriptomics, but to combine multiple levels of transcriptomic, proteomic, lipidomic, and other molecular patterns, along with clinical data, patient-reported outcomes, and lung function outcomes, to produce fingerprints that could be combined to get a much greater selectivity of subphenotypes at a molecular signal level. So the concept is that each individual fingerprint, whether it be a metagenomic or a clinical data fingerprint, will be combined to subdivide these patients to get a much better definition of patients who are likely to respond to disease. We know that asthma is a chronic inflammatory disease with particular types of cells involved in this, and most patients are steroid sensitive. COPD, in turn, is also a chronic inflammatory disease of the airways. However, the particular cell types involved in COPD are slightly different, and the sites of diseases are slightly different. And most importantly, unlike asthma, most patients with COPD do not respond to inhaled or even oral corticosteroids. So there's something different about the inflammation in COPD that makes it refractory to inhaled or oral corticosteroids. We know that cigarette smoke, at least in the Western world, is the major driver of COPD, and cigarette smoke can directly activate both epithelial cells and macrophages and dendritic cells to cause the recruitment and activation of a number of different cell types, including neutrophils, fibroblasts, and CD8 positive T cells. Activated macrophages and neutrophils can release proteases, which are important for the lung destruction, and mucus hypersecretion, which is associated with the disease. As with asthma, this has been a very fertile area for drug development, and there are a number of different drugs or approaches taken by companies to try and develop drugs to target many aspects of this disease. And it's likely that many of these drugs will not be successful in clinic because there are, again, subphenotypes of COPD, as with asthma, and particular drugs are only likely to be successful in these subphenotypes of COPD patients. trying to define these subphenotypes will be as important in COPD by using molecular signatures as it is proving to be in asthma. In one example where we can take a different approach is this study from Dave Singh in Manchester, where they've taken bowel macrophages from COPD patients and stimulated them with dexamethasone to see whether or not there's any anti-inflammatory effect. And the heat map shown here shows that dexamethasone doesn't really affect the inflammatory profile of these cells, nor does the presence of a P38 MAP kinase inhibitor. This kinase is activated in COPD and is thought to be an important driver of disease, but on its own it doesn't appear to have a huge anti-inflammatory effect. But if you combine the two drugs, you can see that a combination of P38 MAP kinase inhibitor and a steroid now start to give anti-inflammatory signatures that may be predictive of therapeutic efficacy of novel combination drugs. And these combination drugs are now in clinical trials for COPD. Another approach, we ourselves have used the proteomic cellsome approach to try and understand why some drugs appear more effective under conditions of oxidative stress, which is present in COPD patients, either as a direct result of cigarette smoking or due to the oxidative stress of abnormal mitochondrial function. And here we can see that in the absence of oxidative stress, in the top panels, steroids are very effective at suppressing cytokine release, but if you give cigarette smoke to cells or you give hydrogen peroxide, the ability of steroids to suppress cytokine release is markedly decreased. However, if you give low subbronchodilator concentrations of theophylline, you start to see a restoration of steroid function. And this is because the ability of theophylline within a cell to target particular proteins and mediate steroid function is altered dramatically by the presence of oxidative stress. On the bottom left panel, we see that there's very little protein association, each protein defined by the little yellow square. But if we have the theophylline in the presence of oxidative stress, we now see a large number of proteins associating with theophylline and the glucocorticoid receptor changing its function. And this gave us confidence to go into clinical studies looking at inhaled corticosteroids with theophylline as an add-on therapy. And this is one study looking at smoking asthmatics performed in conjunction with Neil Thompson and Mark Spears at Glasgow, where we can see that neither inhaled corticosteroids alone nor theophylline alone was able to improve lung function, but the combination gave a significant increase in lung function. And similar data has been reported by Paul Ford and colleagues in COPD patients. So understanding what the mechanisms of drugs by analyzing primary cell data at the molecular level gives us confidence to pick out patients who might respond to drugs. So in summary, I'd like to say asthma is a heterogeneous disease, and it's important to define subsets of patients if we're going to who are likely to respond to new drugs. The random selection of patients without this new biomarkers is unlikely to lead to successful use of these drugs. So we are really looking for a combination of biomarkers or a marker along with a drug to pick out patient phenotypes. And it's quite clear that the use of omics data rather than lung function, which has been shown to be a very poor indicator of patient subsets, is likely to be the way forward in future. In addition, COPD is going to present exactly the same issues that have arisen in asthma, where we have a heterogeneous disease and the need for new drugs, either as add-on therapies to steroids to improve their function, or as new anti-inflammatory drugs to target the inflammation that is steroid insensitive in COPD. Thank you very much.
And thank you, Ian, for illustrating the challenges in asthma and COPD research and the scientific efforts taking place to confront those challenges. We've actually created a kind of roadmap for the presentations that will follow, and I'm sure the presentations will expand on a number of points that you made, as well as points the different presenters are going to be making of their own. If you are just now joining our webinar, welcome. As I mentioned earlier, we will be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Dr. Scott Tebbett, will now begin his presentation entitled Blood and Omics, Can the Mix Provide Insights for Asthma? Scott?
Thank you, John, and thanks very much, Ian. I'm delighted to everyone today, and Ian's given an excellent segue into some of my remarks, but I'll be restricting my remarks to sort of the use of high-performance molecular measurement technologies, as in omics, the use of such technologies in peripheral whole blood, and can we actually discern molecular signatures that can discriminate different phenotypes of asthma. So those are going to be the basis of my remarks today. So my slide is really just an outline of what I'll be talking about. Initially, I'll be talking about the utility of blood itself as a resource for molecular investigations, and also the importance of standardized operating protocols in the collection of these blood samples for sort of high-performance omics analyses. Then I'll be talking about atopic asthma, as well as a specific model, a human experimental model involving allergen inhalation challenge, and the different response types we can obtain following this challenge. Then I'll be talking a little bit about exploratory analyses of plasma proteomic profiling, binding genomic, cellular-based genomic signatures or transcriptomic signatures in the RNA, as well as metabolomic and plasma metabolomic data sets together, and can we obtain more information when we combine information and content across omic spaces. And then finally, I'd just like to touch on one of the main issues with dealing with blood, and that is the cell type heterogeneity present in this complex tissue. So obviously, now moving on to slide, blood is a useful resource for molecular investigation. It's easily obtainable. There's a lot of it in people, and it's obviously the pipeline of the immune system. I draw the attention of listeners to Damien Schelzerbel and his colleagues' very, very readable review in BMC Biology back in 2010. And this is really just following up again on that review in BMC Biology that's basically saying that there's a lot of different technologies and methodologies available to profile the immune system within the blood system itself. And you can see that this is broken down into three different sectors. One sector is genomics, and it can be anything from genetics, looking at DNA from the sequence variation at the base level, right through to epigenetics type modifications, methylation, histone modifications, that kind of thing, through to transcriptomics, looking at the messenger RNA, transcript abundance, even microRNA, and those types of things. On the right-hand side, you can see proteomics. There's a lot of new tools out there now that involve mass spectroscopy and reasonably unbiased approaches to sort of profile the plasma proteome, as well as the plasma metabolome as well. And the lower part, obviously, the cellular aspects, you can do in vitro cellular assays, flow cytometry, and lots of other types of tools available to sort of start profiling blood a little bit more. I'll be mainly restricting my remarks to the genomics, proteomics, and metabolomics space today. Moving on, I just wanted to draw attention to some of the issues around post-collection processing of blood samples. This is very important, especially when you're collecting your samples from research subjects and patients from widely different areas geographically. You might be coming into clinics and different hospitals where you have to combine your samples from various places in order to complete your studies. So one of the three main remarks on this slide mainly is that when you spin out your blood to provide plasma samples, very important to aliquot those plasma samples immediately into separate tubes. So you have multiple aliquots before you freeze them down for the first time. This is mainly because it's been shown quite conclusively now that the plasma proteome can degrade very quickly with multiple freeze thaws. So the importance of multiple aliquoting is very important. On the left-hand upper side of the slide, you see a little lavender 3 ml tube, an EDTA tube, and saying CBC and differential. So we often collect a separate blood sample and send it down for a complete blood count and differential, and I'll be coming back to this issue at the end of my talk today. And lower left-hand side of the slide, this is more about the genomics and transcriptomics. There are a lot of products out there such as Paxgene tubes, Tempus tubes, that have a preloaded solution that in the blood collection tube, that when you pull blood out of the vein into that tube, it stabilizes the RNA content of that tube. This is important for downstream transcriptomic analysis. MicroRNAs are quite stable in peripheral blood when you freeze it and thaw it, but messenger RNAs particularly degrade very quickly unless you stabilize them immediately when they come out of the vein. So in a very nice background in asthma pathophysiology, so I don't want to dwell too much on slide 6 of my presentation, but just to reiterate that it is a chronic inflammatory condition of the respiratory tract characterized specifically by variable and reversible obstruction of the airway. Now, I want to deal specifically with atopic asthma, so where acute exacerbations in atopic or allergic asthma are the result of exposure to allergens. And, through many, many different genome-wide association studies, a lot of genes have become implicated in asthma. So it seems to be, the susceptibility to asthma is driven by multiple genetic factors in interacting with many different environmental factors. So, the sort of unpicking of this is still very much in its infancy, but we do have a lot of GWAS-type data now to sort of gain on that. And as Ian has already mentioned, the whole atopic asthma and inflammation is driven by cross-linking of the allergen to preloaded IgE molecules that are on the high-affinity IgE receptor, and this causes cross-linking of the high-affinity IgE receptor on the mast cell in the lung airway tissue. Then the mast cells release histamine that causes an immediate contraction of the airway smooth muscle, and also releases other mediators, either preformed or through transcriptional processes that go into the bloodstream and start pulling out secondary mediators, mainly through immune cells, and increased permeability through this bloodstream, including eosinophils and in some severe cases of asthma, neutrophils. And in the next slide, I just wanted to introduce this human experimental model that I've been working with, and this is through the Allergen NCE, which is the Allergy Genes and Environment Network of Centers of Excellence here in Canada. And this is they have a subpart of the Allergen NCE is the Clinical Investigator Collaborative, or the CIC. And this group of clinician scientists conduct allergen inhalation challenge on mild atopic asthmatic subjects. And they do so to actually, for early stage 2 drug trials, to see the efficacy of some experimental drugs, to see how well they work in asthmatic or mild atopic asthmatic subjects. So the next slide really just summarizes in pictorial representation what the allergen inhalation challenge is. In the top panel is really showing that the individual research subject who has mild asthma, and will have a whole battery of skin prick testing done on their arm to work out what they're most allergic to, and then through methacholine challenge prior, and then through the Cohort equation, they can actually dial in the dose of the allergen that person needs to inhale in order to drop their FEV1 by a certain percentage. And then they inhale the allergen, and then they record their lung function measurements via spirometry over a number of hours during the day after allergen inhalation. And then the next slide shows typical response profiles after allergen inhalation. So obviously the x-axis is in time, in hours, and the y-axis is the FEV1, the drop in FEV1 from predicted from baseline. And you can see that you get different profiles after allergen inhalation challenge. In the very top profile, you get the immediate early response that can reverse very quickly, and then the subject lung function just continues as normal. In the middle panel, this is quite rare usually in atopic asthma, where you don't get an early response, but you do get a late response, and that is driven by much more of the sort of inflammatory phenotype. And in the lower panel, panel C, you can see this biphasic classical allergen inhalation challenge response type, which includes the early response followed a few hours later by the late response. And it's important in many clinical trials of stage 2A drugs, a lot of the companies are trying to find therapeutics that can actually attenuate and reduce this late response, because that's the inflammatory response. It's very hard to reduce right now. So we've been obtaining our samples through the Clinical Investigator Collaborative during screening programs for subjects to go into clinical trials. And so slide 11 shows some of the data of their FEV1 profiles. There you can see that you can reasonably well separate isolated early responders in the top profile from dual responders in the lower profile here. And you see that they both have this immediate early response, but you get significant differences in their FEV1 drop after about 2 hours, showing that the late response is coming through the dual responders. And we can obtain our blood samples just prior to allergen inhalation challenge, and then about 2 hours after the allergen inhalation challenge. And that's just at the time when the curves start diverging significantly. And slide 12 just introduces the whole process of plasma proteomic profiling. There are many different profiling systems out there, and one of the methods that we've used is called iTrack mass spectroscopy. And the issues with these, compared to genomics and transcriptomics, proteomics is still very much you're not looking at every single protein in your sample. You have a problem with the dynamic range of the plasma proteome. There's huge amounts of protein levels for some things like albumin and the immunoglobulins, whereas the amounts of proteins of the cytokines and chemokines is vanishingly small. So, you can't capture that dynamic range on most technologies. So one of the ways around it is to deplete your plasma proteome of the most abundant processes, and that usually involves antibody columns, and then you do trypsin digests. You can do multiplexing with this with these 8-plex isobaric sample labels, and then you push your peptides that are labeled through a mass spectrometer for measurement. And we've been doing this, and when we do this on a very limited sample set, we can actually find through, when you start looking at methodologies such as sparse partial least squares discriminant analysis, you can actually start pulling apart the dual responders from the early responders, and you can see that the plasma proteome, you can actually find differences in what is happening in the proteome pre and post challenge. And in slide 14 it shows you one particular protein, this is fibronectin, that by iTrack analysis showed at the pre-challenge levels only were higher levels in subjects that went on to have a late response. So in the grey bar, these are the levels in the dual responders at pre-challenge, and in the yellow bars in the isolated early responders pre-challenge. And we've technically replicated this using a different type of mass spectroscopy called multiple reaction monitoring, which is much more quantifiable and much cheaper to run than iTrack analysis. And so this was encouraging because fibronectin has already been implicated in asthma airway remodeling, and that was a paper published by Denise Hocking in Chest. Now let's move on to sort of transcriptomics, or we call transcriptomics genomics, and a metabolomic integration. So you can use technologies such as Affymetrix microarrays, Illumina microarrays. I think there's more and more interest in next-gen sequencing, RNA sequencing systems, to get a fairly comprehensive view of the peripheral blood cell transcriptome in the leukocytes. And when you do this, you can find differential expression between early responders pre and post challenge and dual responders pre and post challenge. And some of the genes that are differentially expressed are represented here by these yellow circles. Now we can also look at the plasma proteome and look for differential expression in the plasma proteome, and these molecules are represented by the blue boxes here. And this is an analysis based on canonical correlation analyses, where you can actually look for correlations, negative and positive, between the transcript levels of particular genes and metabolite levels in the plasma. And what we can see here is that there's a lot more activity, a lot more exploratory analysis, there's a lot more correlations, both negative and positive, as seen by the green and the red lines, connecting the metabolites with the genes. So you can see that a lot of things that seem to be happening in the dual responders who are going on to the late response. And when you pull down a network analysis of this, you can actually see that one of the most significant discriminating features is the enzyme ALOX15. So we can detect differences in these at the gene level, at the transcript level, and actually this is pointing as well to the sort of metabolite level of arachidonic acid metabolism, where you have hits in the metabolome as well. And again, this was encouraging because it was a very limited sample size, looking at blood where we can actually correlate it to sort of the known body of literature that where arachidonic remodeling enzymes are an important factor during eosinophil survival during allergic asthma. So, moving on to the elephant in the room of all these types of analyses in blood, as well as some of the other tissues that you might want to look at, is cell type heterogeneity. And this is the problem here, is that the observed expression of a transcript in a mixed sample from a particular gene is actually the result of the cell-specific expression of that transcript by a number of different cell types. And these different cell types might change in their number relative to each other, and that is what you'd often find in peripheral blood. So this slide just really is a schematic representing that just because a particular gene signal measured by RNA extracted from peripheral blood gives you the same information, doesn't necessarily it can be potentially very likely to be confounded if there are compositional differences between your samples. And, so one person might have more neutrophils than another person relative to lymphocytes or monocytes or even eosinophils. So it's you have to take these types of things into account. And the issue here is that not only can it create false positives in your analysis, but it can also hide specific signals that might be quite interesting, and these would be false negatives. So if you move to, there are ways to deal with these types of approaches without having to go to the enormous trouble and expense of doing more flow cytometry and FACS sorting of your blood sample cells into different bins representing different cell types, and then going to all the extra expense of doing an analysis on the individual cell types themselves. So, this is just a selection of different, more recent literature showing that there are approaches involved either generating cell-specific matrices that you can infer cell composition from a whole gene expression data, or if you have a CBC differential for your particular samples that you have obtained genome-wide gene expression data for, you can deconvolve using linear regression back to those cell type differences between your samples. And that way, when you look at, you can get sort of a little bit more granularity on your data. So this is just a slide showing some analysis of using deconvolution to cell type of asthma blood transcriptome by cell type specific significance analysis of microarrays. And this is post versus pre-challenge. This is not actually looking at dual responders versus early responders. It's just looking at all asthmatic responders here. But you can see here on the series of plots, the FDR, the false discovery rate, is on the y-axis, and the number of significant features at a particular false discovery rate of your transcriptomics is on the x-axis. And you can see, let me just draw your attention to sort of the middle column and the second row from the bottom, and that shows you the sort of deconvolution approach that you can start seeing better false discovery in eosinophils that are upregulated post-challenge compared to pre-challenge. So this is showing you that you can generate gene lists that show significant differential expression that are correlated to the actual eosinophils in a particular data set. We're not saying that the genes are actually upregulated in the eosinophils. We can't really say that, but at least we can say that they're correlated. And so these are types of approaches that are fairly promising future initiatives that we can get some more traction with. So in summary, hopefully I've convinced you that peripheral blood can actually be a useful resource for molecular investigations, not only asthma but also other inflammatory disorders of the respiratory system. I would recommend that if you can get, again building on some of Ian's remarks, exquisite clinical phenotyping is absolutely essential, especially for omics-based profiling, where you're dealing with large numbers of effectively statistical tests and usually with low numbers of subject samples. Integrating the omics data sets actually may lead to more comprehensive understanding of the processes going on, and also, finally, statistical deconvolution approaches can help to deal with blood cell type heterogeneity in mixed samples. And just finally, a slide of acknowledgements, and thank you very much.
And thank you, Scott, for that fascinating presentation. on using blood as a resource for molecular investigation with omics methodologies. I'm pretty sure many of our webinar attendees are going to take your major points of discussion and incorporate them into their own research projects, so thank you very much. Before we proceed, let me remind everyone of our Q&A segment that comes right after the panelists have made their presentations. Please type your questions into the Ask a Question box on the lower left of your console and then hit Submit. Our third panelist, Dr. Yuri Nikolsky, is ready to describe the MetaMiner pathway analysis platform for research on asthma. Yuri?
Well, thank you, John, and thank you, Ian and Scott, for very interesting presentations. I would actually like to start with Scott's talk. He applied a number of methods of pathway and network analysis and omics data for his research on blood and asthma, and that's quite typical. So any complex disease now requires omics data analysis and very different aspects of analysis of this data. So analysis of omics data requires quite special tools of pathway and network analysis and the disease knowledge accumulated in a certain database in computer-readable form. And I would like to describe what we've done for this over the last 2 years. It's our development called MetaMiner Respiratory Diseases. And I will also show a couple of brief case studies on how the system can be applied for analysis. So the next slide shows that you probably don't need to have any disease knowledge at all if you have well-working statistical method on omics data. So essentially you can come up with gene signature or multivariant biomarker pretty much for any kind of phenotype, for drug response or for disease subtyping. The problem is they are very different genetically, and this high heterogeneity prevents from using just gene signatures for biology. Otherwise, pretty much all the disease would be treatable by now. So this slide shows the heterogeneity. So you have 2 different patients, so you can have 2 different studies, and the top genes actually can be quite different. However, these genes can still be in the same biology, can share the same molecular network, or like in this case, to belong to a certain pathway. So if in such case, the genes per se are probably not very powerful as a biomarker, but the pathway itself becomes a biomarker. So that's pretty much the reason for us reconstructing disease biology. So the next slide showed that deep understanding of biology is actually important for pharmaceutical industry. So the critical mass of biological information is important for better return on R&D investment, for better hit rates and drug discovery in general. And patient stratification based on biology is really growing and important, and essentially most of new drugs, if not any, have to come with certain biomarker reflecting biology of drug response. So, however, in the next slide, you can see that you really can represent biology in very different ways, and different groups and different fields studying biology sometimes can actually talk very well with each other. So molecular biology, biochemistry, clinics, they can even use quite different language and semantics. So these were the issues which we had to deal in MetaMiner program. So why MetaMiners and how we see disease reconstruction? For us, reconstruction of any disease, and especially complex diseases, asthma, has 3 parts. So one, we need to reconstruct disease pathways, pathology pathways, which are quite different than normal biology. That one part. Another is annotation of pretty much all the changes, all gene alterations which are associated with a certain disease or a certain onset of disease. So these experimental biomarkers at RNA, protein, and metabolic levels. And finally, it's a repository of omics data, NIF data, and expression data done for the disease. So on the next slide, we show that we've done that before. So the program on disease reconstruction started with cystic fibrosis, the Cystic Fibrosis Foundation about 5 years ago. And then we completed a number of projects on cancer, CNS diseases, skin toxicology, stem cells, and so on. And several programs are ongoing, and one of them is on asthma and another on COPD, and we do it with major pharmaceutical companies. So the next slide shows the main deliveries of this partnership program. So one is a set of pathway maps, and in case of asthma, it's more than 60 maps by now. So they use mostly for enrichment analysis of expression, SNP, and other omics data. Then it's a large set of biomarkers at RNA, protein, DNA level, and a combination of pathways and biomarkers. And finally, the annotated and normalized data ready to use in repository. So the next slide describes how we do it. Essentially, it's our annotation technology. We've been working on our own annotation technology for more than 12 years. It's really hard work, so we have more than 70 scientists, PhDs, and MDs, mostly in Europe, and some of them are in India, and they just read journals, more than 3,000 journals, more than 400,000 articles altogether. And then they use our technology for recording certain type of data according to our semantics. So this is really a database which can be retrieved and used in different ways. We call this database. And this is the background for our additional work on disease biology. So the next slide shows the complexity of asthma as a disease, which is represented in the form of pathway map and schema. So asthma is inflammatory disease caused by chronic inflammation in airways, and this inflammation leads to airway obstruction and bronchospasms. So epithelial layer is altered in asthma patients, and it's characterized by shedding of epithelial cells, large number, and hyperplasia of goblet cells, mucin production, and eventually immune response. So what's important here is that there are many different types of cells involved in asthma pathology. So we deal with goblet cells, epithelial cells, smooth muscle cells, parasympathetic nerve, and a number of blood immune cells such as eosinophiles and neutrophils. And all these cell types are interconnected by pathologies and certain mediators like interleukins, and all that has to be described in very specific ways. So we need very specific pathology map for goblet cells, for epithelial cells, for blood cells, and that has to represent state of art in different fields. So that's why it takes so long. So we work on this project for about 2 years and probably continue for another at least 1 year. We drew 25 maps for lung epithelial cells, 13 maps for smooth muscle cells, maps for fibroblasts and mast cells. Altogether, it's about 60 pathway maps. And currently, mostly we work on immune cells, T cells, macrophages, eosinophils, and neutrophils. So we believe that will be another probably 30 pathway maps in addition. So next slide shows some sampling of epithelial cell maps. You can see that the maps are quite diverse in biology they cover. So we cover disruption of apoptosis and proliferation epithelial cells, Th2 and TNF alpha, TGF beta mediated process and expression changes, and downstream biology, induction of EMT by BMP4 and TGF beta 1, and many other aspects of biology. So the next slide shows how we actually draw these maps. We start with interactions, and interactions are stored in the database, and new ones get annotated every day. So interactions define one step in cellular pathways, signaling pathways, and usually they come from very different experiments and different studies. So when we put them together, it looks like a chain of signaling interactions and metabolic reactions. And then we read quite a lot of literature to confirm the pathway as a whole. So the next few slides show some examples of our pathway maps. So on the next slide, it's about one of the most important pathway maps for asthmatic airway epithelium, and it describes interleukin induction of expression of pro-inflammatory cytokines by epithelial cells. So the most important interleukins are IL-33, IL-1, IL-1 alpha, IL-13, and IL-17. And you can see on top where these interleukins come from. So from different cell types participated in the response. Then there is epithelial cell itself. So it shows actually signal transduction from cytokines down to transcription factor, and eventually induction of pro-inflammatory cytokines such as TSLP, IL-8, and CCL5. And downstream is pathology caused by increased levels of these cytokines. So the next map is more detailed, and it described BMP4-mediated EMP induction in epithelium. So this is a process which is kind of counterpart of TGF-beta induced phenotypical changes, and it describes different effects of BMP4 on expression of other genes, mostly transcription factors in nucleus. So the next pathway map deals with a different type of cells involved in the response. So these are airway smooth muscle cell, and it describes proliferation in case of asthma for two main reasons: increased input and elevated level of calcium, it's on the left, and also decreased expression of CBP alpha, which normally inhibits proliferation, that's on the right. So what is shown here, the way the maps actually connected, so these purple squares actually gateways to other pathway maps which are related to this pathway map. So the maps come as a set, so you can switch from one to another, and when you do enrichment analysis, actually they come out as a ranked set of pathways. So the next slide shows the way we annotate biomarkers, and by biomarkers we understand any change, any alteration at the level of epigenetic or genetic RNA happening in any gene which is related to a disease. So at the level of DNA, we cover methylation, histones, different kind of aberrations, somatic mutations. Mostly, of course, it's polymorphisms, SNPs, gene variants. Level of RNA is mostly abundance, higher level expression, and sometimes it's splice variants. Proteins, to a large extent, it's PTMs, phosphorylated proteins in case of diseases. Also a change of localization and change of protein level. And in case of metabolites, usually it's about higher and lower concentration. So the next slide shows the number of genes we annotated for asthma. So this is a fragment of MASH disease tree. And for asthma, we have 900 genes, 908 genes, which would have any number of alterations. And altogether, we have about 4,500 biomarkers in these 900 genes. So the next slide shows a snapshot of our SNP representation for asthma. So you can see one of the genes here called ORMDL3. It's one of the new genes, just 3 genes family dealing with protein folding, and quite a number of SNPs actually have been identified in them. In some cases, we know that the SNPs cause gain of function, so that's the third column for activity. It's shown that it's up. Then it shows the cell types, so most of these specific biomarkers are in leukocytes and other blood cells, and then there are references for each of them. So the next slide shows what we cover in the reference. It's a very short abstract, the description what was shown in the paper, the patient cohort and disease. In this case, it's French Canadians, 253 people in cohort, and it's asthma without any subdivision. Then it describes SNP and what happened as a result of this SNP. We also annotate drugs, so altogether we have 276 single-agent drugs and 32 combinations, and most of these drugs are experimental in clinical trials, phase 1 through 3. So the next slide shows how everything is put together. So here we have normal pathway maps ranked by these 4,500 biomarkers associated with asthma. So this is like a standard set of our pathway maps, about 800 of them. And you can see that mostly immune response cells would be disrupted by the biomarkers and populated by these biomarkers. So the top map would be immune response for Th1 and Th2 cell differentiation, and then a number of other immune response, mostly in blood cells. So the next slide shows what actually the meta-minor maps and meta-minor disease reconstruction adds to this picture. So now we have a very different type of cells which normally are not described in pathway databases. So we have mast cells, we have asthmatic epithelium, fibroblasts, smooth muscle cells, and so on, and very specific pathology which is populated with asthma biomarkers. And you can see with the p-values that the p-value is very low. So for top map it's e to the minus 26, e to the minus 17, which shows relative importance of this biology for asthma as a disease in general. So the next slide shows how these biomarkers can be represented on pathway maps. Actually, they can be quite nicely visualized. So this is one of the fibroblast response maps and it shows like mechanistic part of this response. So there are genes and some compounds. They're connected through by protein interactions. So each interaction would lead to a reference, and this is a mechanistic view. What we can do is just like to go to the software called Metacore and look for diseases and try to visualize all kinds of biomarkers on these pathway maps. And I selected here all levels of biomarkers: RNA, protein, DNA, and metabolite compound. So for protein level, for instance, we look at decrease of protein activity, increase of protein activity, high abundance, isoform phosphorylation, and so on. So all kinds of alterations. So the next slide shows a fragment of the very same pathway map in what we call causal view. So now we have the genes which are altered in asthma highlighted, and different letters and squares show what exactly happens here. So for interleukin-4, for instance, there is a SNP in the gene. It's related to disease, and also it's both expression level and protein level are increased in case of asthma, and so on. So quite a number of genes in this particular map would be associated with asthma biomarkers. So now I would like to show a couple of examples how this environment, this platform, MetaMiner, can be used for data analysis. This is a quite straightforward case. One of the studies published last year by a team from Genentech, and they looked at the gene expression patterns in response of Th2 3-gene signature in asthmatic airways. So essentially they looked at asthma versus control, high 3-gene signature versus low T-gene signature, and one of files was drug response versus control in asthma. So the next slide shows enrichment analysis in respiratory MetaMiner and the top pathway maps. So you can see that out of top pathway maps, 8 would come specifically from this pathology map development, and they would deal mostly with epithelial cells, which is logical based on what this experiment was about. What's important here is that you have most of these pathway maps coming through full discovery rate filter of 0.5, which is not always the case when you do enrichment analysis. If you look at the next slide, which is plain Metacore, so here we excluded any of asthma-specific pathway maps, you see some changes, like in cell adhesion, for instance, cytoskeleton response, and so on. But many of these pathway maps actually would not be statistically significant even. And you can see that they're not specific enough really to describe the changes which happened in specific cell types involved in asthma. So the next example deals with drug repurposing and using the MetaMiner as a tool to suggest EGFR as a potential potent drug target for asthma. So in this case, we applied several approaches based on knowledge management to assign EGFR as a new target for asthma. EGFR, of course, is a very well-known drug target for different cancers, but not really the main target for asthma. So the first method assumed that if gene belongs to a set of biomarkers for a disease, it gives it a certain score, so it can probably be used as a target. Not like complete evidence, but it's part of the evidence. So EGFR is a known biomarker for asthma. We annotated it with a number of SNPs associated with disease onsets. Then we look at the interactions of EGFR and the first level of interactors, proteins connected to EGFR within like one interaction level. And some of these genes, like ZO1, is also a biomarker for asthma. So it's another score in set of evidences for EGFR. So another approach is to look for neighbors on normal pathway maps, and this is one of the maps. Role of interleukin-8 in angiogenesis, and EGFR is situated on the same pathway map as HMDH, which is well-known drug target for asthma. So the next method actually uses specific MetaMiner pathway maps, and you can see that there are at least 12 different maps where EGFR belongs among the genes. And the next method, actually derivative of the pathway maps method, it just looks at specific signaling pathways, linear signaling pathway where EGFR belongs. And the same pathways would contain ethylene, and this is a known drug target. So put all this evidence together, give us a pretty high score for EGFR to be to be a novel drug target for asthma. And this is a page from Integrity, our database of drugs and bioactive compounds. And it does show that there is at least a couple of compounds which are experimental, which can be now tested for asthma therapy as experimental drugs. And this are a couple of references from this year and last year showing that indeed EGF receptor can be used as new asthma target. So to summarize, we believe that understanding of disease biology is really key in modern drug discovery, and quite a number of researchers share this view. We started working on reconstruction of diseases quite some time ago, and by now we have good technology for this non-trivial process. So metaminor asthma is a 2-year-old program, and it's quite advanced. So we can reconstructed over 60 disease pathways of asthma, and in parallel the development for COPD, it's about like 10 pathways by now. We have about 4,500 of experimental biomarkers for asthma. Mostly these are SNPs, but they cover actually RNA, protein, and DNA level. And we have quite well annotated and normalized repository of asthma expression data sets, which can be used for comparison with customer data sets. And well, application of this program, this development, pretty much everywhere in drug discovery can be used for patient stratification, personalized medicine, drug repositioning. Well, thank you very much.
And thank you, Yuri, and thank you, Ian and Scott, for providing our attendees with a host of original insights on new ways of conducting asthma and COPD research. You certainly made an impact on our audience, as we've received a number of great questions from our attendees for our Q&A session. Let's take the first one. And Ian, the question, number one question is for you. What is the status in research of reversion of disease symptoms and cell types when using stem cell-like treatments?
Okay, I think where we are at the moment is really in animal models of asthma. So people have looked at both inducible and bone marrow-derived mesenchymal stem cells as a means of trying to skew T-cell function in these animal models of asthma. And there you can see quite nice evidence for reversing the changes in the airway hyperresponsiveness and inflammatory mediator responses that we see. So I think currently in asthma, with preclinical models, it looks interesting in those data. I haven't seen any evidence in COPD in relation to these models, but it wouldn't surprise me if we didn't have similar sorts of studies ongoing in animal models of COPD. It's just that I'm not aware of them. So I think we're going down that line and sort of watch this space, I guess.
And Ian, we have another question. What is the opinion on the role of environmental mold and control of conditions to help in prevention and remediation to counter the effects leading to asthma and related afflictions?
Okay, so aspergillosis, for example, is well known as a factor that has been associated with severe treatment refractory asthma, and removing sort of infections from these fungi clearly makes a difference. If you treat the infection, then the patient improves. If we look more generally at other environmental agents, whether we're looking at house dust mite or cat dander, for example, then clearly we can see that removal of these drivers can make a marked difference to the quality of life of these patients. They no longer exacerbate. Although saying that, I mean, if someone with a severe asthma who has a cat, it's incredibly difficult to get them to get rid of the cat. They'd rather have severe asthma being hospitalized 3 or 4 times a year rather than get rid of the cat. So sometimes it's difficult to remove the environmental stimulus. But I think it is important to remove, if there's a known environmental driver, to remove these as much as possible before we go on to using more and more stringent drugs that have potential side effect issues.
Actually, and it was very interesting your point about the cat. I had a friend who had a cat for about 16 years and suffered from horrible asthma, and it only stopped when the cat finally died. So
so true.
That's what they do. Hey, Ian, we have another question here. What is the status of the various PDE4 drugs, and do you see these drugs as being effective in the treatment of COPD patients?
Okay, so PDE4 inhibitors, such as roflumilast, are currently on sale. They're on the market in Europe, or various countries in Europe. And their prescription rate varies depending on the country you're in. So they're fairly high in Germany, but low elsewhere. They have minimal effectiveness, at least in the phase 3 studies, where they're looking at an improvement of about 48 or 49 mils in FEV1. And I think their role as a monotherapy is not going to go forward. In combination with steroids is maybe another matter. You may be able to target an inflammatory component that is not hit by steroids and therefore get improvement. I think one of the issues still with the drugs on the market is we haven't totally dissociated the problems with diarrhea and vomiting from most PDE4 inhibitors. And this is an important issue if you're a 60-year-old with quite a severe disease such as COPD. So I can see that there are issues. They're on the market, and I think the market will tell us how well they're doing in the next year or so.
And Ian, what specific biomarkers do you recommend for measuring in airway epithelial cells?
Well, I think what I would say is that if we're trying to look at steroid responsiveness, there's some wonderful work done by Prescott Woodruff and John Fahey, which really have used unbiased gene array approaches and then finally pulled down into looking at a 3 mediator signature for those that will suggest that these cells and patients presumably will respond to steroids. And this is the periostin. There's SERPINE2B and CLCA1, so a marker of innate immunity. So I think there are specific biomarkers that people have now published on that give us an idea of the responsiveness to particular drugs. It would be nice if we had a number of other signatures relating to other drugs, how they affect these cells, but these aren't available to my knowledge.
This next question is really great for those of us who live in Manhattan, where we see more cars in one day than some towns see in probably a year. Is there a correlation of asthma incidence and the concentration of tire dust in the air?
Okay, tire dust is difficult to define in itself, but I can tell you about the studies looking at traffic pollution or air pollution in general. So clearly, studies from both US and from Europe indicate that when traffic pollution is higher, then people with ongoing respiratory disease get worse. So, and this has been linked with in studies from Antonio Baccarelli in Harvard would suggest that when you go away or you don't go into work over the weekend, you improve a little bit, and then it takes a couple of days after sort of coming back to work on the Monday, Tuesday that you start to feel worse again. We actually did a study in the UK from the Brompton Hospital where I work, where we put asthmatic patients and we walked them up and down Oxford Street, which is really just buses and taxis, and compared that with walking around Hyde Park, which is only about sort of 400 yards away, but is a big open space in the centre of London, and it makes a huge difference if you're an asthmatic walking down Oxford Street. You immediately have a drop in lung function, or at least as quickly as we could measure within half an hour, and you get an increased release of inflammatory cells into your sputum and inflammatory markers. So there's quite pronounced effects of obviously traffic pollution, and this will be diesel fumes. I'm not sure if anything's been done directly related to tyres, but if tyres has any relationship to the amount of pollution that these cars are spewing out, then it wouldn't surprise me.
Well, Ian, I get to London a couple of times a year, and Oxford Street is definitely off my list now, so thank you for that practical advice.
All right.
Hey, so Scott, we have some questions for you. Did you make a comparison between PAX gene and Tempus extraction?
Yeah, thanks, Ian. Thanks, John. We haven't directly compared Tempus tubes with the PAX gene tubes, but I believe other people have, and it's in the literature. You might want to check under Dr. Tim Trishes at the Los Angeles Children's Hospital. I think he has compared those two tubes. I would only like to reiterate for this question is that there's a lot of controversy over whether they're any good or not, because they are quite expensive. They can be up to like $10 per tube, which can add up when you have a reasonably large study. But the really important thing is to make sure they're mixed carefully and thoroughly, these contents of these tubes, with the fresh blood immediately on blood draw before you put them in the freezers or on ice or anything like that. And I think as long as people carefully follow the protocols, because often these are FDA cleared as a kind of a clinical diagnostic apparatus, it's very important to follow the guidelines of use of these tubes. People, especially in a busy clinical laboratory, a lot of the research nurses have got looking after the patients, and they've got other things to worry about. But it's really important to sort of definitely get the blood correctly into that tube and mixed.
And Scott, someone wants to know: are the early responder genes a generic inflammatory signature, or are they specific to the allergens?
That's a good question. In our experience, at least on a smallish sample size, the early responders, there's a preponderance of neutrophils in the early responders at pre-challenge, and that can actually, when you actually look at the gene expression from the whole blood, you obviously get inflammatory signatures there. However, when you take into account using a covariate analysis, you then find genes that are more cell cycle type genes that are activated. As far as the different allergens are concerned, when we did our study on early responders versus dual responders, we restricted it to individuals who had been inhaled cat allergen, mainly because it's reasonably well established now that the use of the different allergens do give you different responses. There was a paper, I think a year or two ago, that challenged people with house dust mites, and that generally gives you a dual response, whereas if you challenge people with grass allergens, that can give you predominantly isolated early responses only. So in our cohort, we find that cat allergen can give you sort of a reasonable mixture of either isolated earlies and dual responders. So in our initial study, we restrict ourselves to the cat allergen.
Okay, and boy, we really got some great questions here today. Scott, are there recommended resources on blood cell-specific biomarkers that one can apply to deconvolution of the blood omic profiles?
Yeah, another great question. In my slide that sort of summarized the sort of literature out there, the recent literature, I'll probably just draw the questioner's attention to two particular papers. They're both in PLOS, Public Library of Science, PLOS 1 journal. And one is by Gong et al., Ting Gong, and from Novartis Group, and the other one is from Alexander Abbas from the Genentech group down in San Francisco. So, both of these organizations or groups have used either gene expression profiling of specific cell line RNA, which have been obviously immortalized, so they might not necessarily be perfect reflectors of what is actually happening in peripheral blood. Another
another group they used FACS sorting to pull out different types of blood cells and then did transcriptomics on those individual cell types, which might be a little bit close to what's actually going on in the blood, assuming the cell sorting itself doesn't affect the gene expression profiles. Now, what either of these organizations have done is show that there are specific gene-specific signatures in each cell type, but the trouble is it's very messy. So, even looking at T regulatory cells where FOXP3 is a really nice specific gene profile. However, FOXP3 is also expressed to some extent in activated T cells. So, depending on your question, you're trying to discriminate Tregs from activated T cells. When you're just looking at the RNA, it can be problematic. But I would certainly recommend looking through these papers and looking at these sorts of things. The problem is that you probably need to use at least a dozen, 2 dozen, even up to 50 different genes that are reasonably specific to a specific cell type, because it's virtually impossible to find genes that are only expressed in one particular cell type. So you're going to have to use a multi-type of approach to do this. So, it's, as I say, it's kind of early research in this area, and whether microarrays have got the dynamic range to really drive this forward or not, or whether we need to move over to RNA sequencing, which has greater dynamic range, I think that's still an open question, but it's an active area of research.
And Scott, phenotyping is critical for asthma genetic association, according to this investigator. Could you discuss the role and type of endophenotypes as best predictive of asthma?
Well, it's another excellent question that sort of gets to the point of what is asthma. I think with Ian's remarks and some of the things that I've been talking about with these early and late or early and dual responders. It's pretty clear now that asthma represents a very heterogeneous mix of different disorders, effectively, with underlying differences in susceptibilities and environmental impacts. So I think the question is absolutely right to have good endophenotyping to identify which groups you're going to put these people into for genetic analysis. Again, the problem is with, if you take GWAS or genome-wide association studies, in order to power these studies, you're going to have thousands of subjects, asthmatics in one group and non-asthmatics in the other. And the problem is, what they've been finding up until now, at least, is that some of the relative risks of particular SNPs or genes are very low. So you're only really getting 1.2, 1.3 relative risks for any individual genetic marker. So the problem is you don't have the penetrance of these markers, at least for asthma, to get into this on when you have a phenotype just asthma. And often these phenotypes will just be called doctor-diagnosed asthma. And some of the questions you'll have for the recruitment into these studies are: have you ever been diagnosed with asthma? So that's not very good. It's very good to find lots and lots of subjects for your genome-wide association studies where you're testing 500,000 SNPs, but it's not very good to get into the endophenotypes. And so I think these other approaches are very nice if you can actually show these endophenotypes. But the problem with that is that you're never going to get very many large numbers of subjects in each of your buckets to ask your genetic questions, certainly not at a genome-wide level. So it's an excellent question, and I think it will be interesting over the next 5 years how far we can get in with the genetics.
Thank you very much, Scott. And Yuri, we have some questions for you. What is the difference between a network and pathway in MetaMiner?
Well, we have two different ways to visualize protein interactions in MetaMiner and MetaCore. One is the pathway maps and another is networks. So the pathway maps are kind of a static representation of relationship between the genes, and they built by our annotators. Sometimes they spend 1 or 2 weeks on each pathway map. so they don't really change. Well, there is editing tool which allows you to be allows you to change anything, but usually people use them as is, so they're kind of standard. We also have pathways, and by pathways, signaling pathways, we understand linear sequence chains of protein interactions, each one of them experimentally confirmed, and 11 steps on average. But this is something which is preset by our annotators' best attempt to describe biology. So the networks are built also out of protein interactions in the database, but they are usually unique for the data set. So you have your expression profile, differentially expressed genes, so you can have mutated genes or SNPs, and use that as seed nodes, and then can apply different algorithms to build a network out of interactions. So those are kind of different applications. So pathways are usually used for enrichment analysis at the first step, and networks is a bit more advanced tool to go into details of what you have in your data set.
And Yuri, a question, how frequently is the MetaMiner database updated?
Well, the underlying interactions database, which we call Metabase, is updated daily on our internal servers and quarterly for the customers.
Okay, and another question related to MetaMiner. How do the MetaMiner maps account for the influence of heterogeneity in cell composition raised by Dr. Tabut to be predictive for people?
Well, Scott's example was mostly on blood cells, and we haven't covered blood yet, so it's upcoming. So next year we're going to cover T cells, macrophages, eosinophils, and neutrophils. So then the tool will be quite well representative for heterogeneity. So essentially, when you have a mix of cells and very specific sets of pathway maps which are specific for a certain cell type, you have a certain distribution of p-values, and this distribution of pathway itself can become a descriptor for a certain type or subtype of disease. So then it's well applicable, and we've done this kind of analysis when the distribution of cell-specific pathways become a descriptor for different cancer applications, and people use them for microbiome analysis. So essentially you can use the p-value distribution as a certain characteristic for a subset of asthma, and quite well applicable for drug response evaluation as well.
And Yuri, are these disease signaling pathway maps publicly available?
Well, not really. They're available through our software, which is called MetaCore. So some of pathway maps which are off exclusivity period available right now, and some other available only for members of MetaMiner consortium.
Okay, and then Ian, coming back to you, we have a question. What is the greatest limitation in using animal models in environmental exposure studies?
Well, I think the issue with animal models, both in asthma and COPD, is not just related to environmental models. I think it relates really to the fact that they're not such good predictors of disease efficacy. So many drugs will work in animal models, whether we go from sort of rodents, mice, rats, we go to guinea pigs, or even non-human primates. They're not necessarily predictive of human disease, which may reflect the phenotyping or the endophenotypes that we've just been discussing. So I think the limitation is really having good knowledge of what our model is really modeling. And for asthma, that's been a particular issue. There are no currently available good models of asthma severity, for example. We can't get good models of exacerbation of asthma. And if we go to the environmental aspects of COPD models, most people believe that cigarette smoke is a good model of COPD, because we know that in the West cigarette smoke drives the disease. But depending on how you expose the animals to cigarette smoke. It could be side stream smoke, or you could pull the smoke through the filter. It goes through a tube, it goes into a big tank or whatever, into a fairly large exposure chamber, which the animals then breathe. This is not the same as us smoking. I mean, you can see models where, or in at least for the smoking models, there are monkeys in some countries that are taught how to smoke. That may be a better model, but it becomes very, very difficult in trying to get good environmental exposure models in animals that mimic really what goes on, when, particularly the way we breathe versus the way an animal breathes. So it's a limitation of all the animal models rather than just of the environmental aspects. But it may be that, say, acute over challenge, for example, is a very, very good model of the classic steroid-responsive asthmatic, but we don't get the subphenotypes of asthmatic out of that.
Well, thank you very much, Ian. Unfortunately, we have run out of time. Please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. I want to say thanks again to the panel for their outstanding presentations, and I want to say thank you to our audience for your attention and for your thoughtful questions about the various topics brought up during the webinar. And thank you to our sponsor, Thomson Reuters, who made this event possible. Shortly, we'll be sending you a survey, and we would appreciate your feedback on this webinar. Please look out for it, and kindly give us your thoughts, as this will help us to continue to bring you topical and timely webinars in the future. Thanks, and have a great day or a great evening wherever you are. Bye-bye.